Welcome to the lecture series in food microbiology. In this series, we have been dealing with the various microbes which inhabit the food, the beneficial ones, the ones which cause spoilage, the ones which cause diseases, then how to prevent the diseases or how to control the growth of microorganisms in food. Among the ones which cause diseases in humans because of fecal-oral route of contamination, one major aspect which we are discussing are the parasites. These parasites, generally they are the nematode worms as we call them, are shed in the fecus and produce infection in those who ingest them with food and water. Some of the other parasites are present only during a portion of their life cycle in animals used for food, and subsequently these are eaten by humans, whereupon they carry out the sexual reproduction in the humans, produce eggs which are shed along with the fecus. And none of the parasites transmissible through food or water is capable of multiplying outside the host. Worms, the roundworms, which are the common names, they are parasites that live in the intestine. They have a long round bodies and range in size. These are usually found in soil and stool and can enter the body through the mouth or direct contact with broken skin, and they can live and survive in the human intestine for a very long time. There are several kinds of roundworms and all of these are equally and quite harmful. The life cycle of the nematodes may occur entirely within a single host, or it can be more complex involving multiple hosts. Reproduction is normally sexual and the female may either lay eggs or give birth internally. So, this is a picture of the common roundworms which are seen in the intestine. One major example is Trichinella spiralis. The transmissible form of Trichinella spiralis is the larval cyst, which can occur generally in pork muscles. So, people who feed on pork are the ones which are not with the animals which have been infected. They will carry this cyst in the meat or in the, and when those are consumed without proper cooking and processing, people will get infected. This is sometimes referred to as the pork worm due to it being typically encountered in undercooked pork products. And generally how to safeguard from this is that pork should be cooked till there is no pink color visible in the meat. Once swallowed the larval cyst, the adult worm burrows into the lining of the intestine. Cysts that are eaten with meat liberate the larvae which undergo sexual differentiation. The adults penetrate the mucosa of the intestine to copulate, and later the males re-enter the lumen of the intestine and then are carried out. The female burrows deep into the mucosa and produces approximately 1500 larvae, and the newborns are carried via the lymph and the blood to the striated muscles where they grow and encyst. This is the picture of the cyst, the Trichinella spiralis in the human muscle. The larvae burrow into the striated muscles, causing edema, muscle and joint aches and fever. And eventually, the high concentration of the cysts in the striated muscle will result in the loss of efficiency of muscular contraction. In people who have died of trichinosis, more than 1000 cysts per gram of the muscle have been found, which is a quite huge number. If you see the life cycle of this is the normal cell larvae which is present. The larvae are ingested, for example, in the undercooked meat they are ingested and they are in the intestine, and once from the nerve cells these are now liberated out and the larvae reach the small intestine. The adults mature and they live in the small intestine of the host or the humans, and then there you have the newborn larvae which are present here. The female sheds the newborn larvae that enter the lymph or enter the blood. They can carry it through the bloodstream to the other striated muscle cells, and if so, if you see in the pathology, if it gets into the brain you have the CNS damage, if it gets into the heart muscles you have the heart failure here. The newborn larvae enters the skeletal muscle cells and then they mature in the muscle. See, this is the larva which is maturing inside the muscle, and then again you have a complex which is formed which gets liberated. So, the cycle will continue like this. Diagnostics. Trichinella spiralis is the most important species in domestic swine for zoonotic transmission. For the detection of this parasitic infection, two primary methods are used, and basically swine testing is the one which is done. The preferred method for diagnosing infected swine is by digesting the meat samples or any sample. Suppose you have bought from a farm some amount of swine meat and you want to test for the infection. You digest the meat sample, for example the diagram, the eater's tongue or any part of the muzzle with HCl pepsin. This protocol was devised by Ribic et al. 2001. And then you look inside the muzzle. Once it has been digested, you look for via microscopy for the presence of the larvae. Though cumbersome, sensitivity of this method is relatively uniform throughout an infection. Though it is laborious, you can visualize the larvae under the microscope. Enzyme-linked immunosorbent assays, or otherwise which we discussed earlier, ELISA, is an alternative method that provides for the relatively rapid detection of spiralis infection by serology. And one advantage is that ELISA can detect infection even prior to slaughtering the animal. There are several tests available to diagnose trichinosis in humans. One is muscle biopsy. This is used to directly observe muscle larvae for unequivocal diagnosis. So, once a person's muscle has been seen to contain the larvae, then the infection has been, it's positive. Biopsies can be evaluated by several methods involving microscopy. One is pressing tissue samples between two slides for detection of muscle larvae in tissue. Second is digesting a finely minced portion of the biopsy sample in HCl pepsin, and then you detect the muscle larvae released from the tissue. Histological examination of the stained sections of the biopsied muzzle. Along with these microscopical techniques, several serological assays have been used for diagnosing human infections, including indirect immunofluorescence assay or IFA and ELISA. But the problem with IFA and ELISA is, along with the infection of trichinosis or this, it can interfere if the person has other parasitic infections also, then there can be cross reaction, thereby giving an incorrect result of the scope of the infection. This is the negative aspect of IFA and ELISA with regard to diagnosis of trichinosis. The second parasite we are concerned with is Ascaris lumbricoides. This is normally a small intestine dweller, what we commonly call as the worm in a human is normally Ascaris. So, the adult worms, there is a sexual dimorphism exhibiting between the male and the female worms. They normally, the eggs or the larvae are present in the soil and can get inside through skin also, and they can be contaminants on fecal-oral route. These are the large parasites that can be transmitted in the form of eggs. The eggs of these worms are found in sewage, fertilizer and in soils. The eggs may contaminate crops grown in soil or fertilized with sewage. Infected food handlers may contaminate a wide variety of food. People who consume uncooked vegetables and fruits grown near soil, in or near soil fertilized with sewage are vulnerable to the infection. Eggs of Ascaris have been detected even on fresh vegetables. Infections may self-cure after the larvae have matured into adults, or if the infection is severe, it may require antihelminthic treatment. And in severe cases, surgical removal may be necessary. There have been well published newspaper reports about the amount or the mass of the number of worms which have been removed from children after they complained of pain, nausea and show generally malnutrition tendencies. Allergic symptoms are common in long-lasting infections or in reinfection. The worm is pathogenic because the larvae leave the blood and move into the lungs, causing hemorrhages and pneumonia. The worms can also access the pancreas and bile ducts, producing obstruction. This will happen when the load of the worms or the parasites present in a person becomes huge. So, the step 1 is like, if you see, you have the fecal-oral route, the ingestion of the embryonated eggs by the human being. So, then what happens? The eggs pass on So this is the adult in the small intestine of the human being. Now, what will happen here is that this is the larvae are coughed up, swallowed, re-enter the gastrointestinal. This happens in the case of a reinfection when it gets circulated inside the human body. So, once the embryonated eggs are ingested, in the intestine you have the adults. These present in the small intestine. Now, this is here, the adult worms are here. So, these now will produce eggs. The fertilized egg is shed out. The unfertilized egg will not undergo further development. This is passed out via the fecus. The fertilized egg now becomes the infective stage. This is now outside in the body, and the hatched larvae, this is the egg with the L3 larva, which now goes back into human for a reinfection. Now, the second category what we are talking about is from the intestine of the person, infected person, the hatched larvae can now under the circulatory system and migrate to the lungs. Now, this will now inside, so this is now migrating to the lungs here, and from the lungs when you cough or when they are again swallowed and they go back to the GI tract, and then these larvae again reach the intestine where they will mature. So, this cycle of reinfection will continue, and finally the larvae reach everywhere in the all the cells of the body. So, it becomes, so this is the diagnostic stage, this would be the infective stage of the infection. The adult worms live in the lumen of the small intestine. A female normally may produce approximately 200,000 eggs per day, which is quite a huge number, which are passed out with the fecus. The unfertilized eggs may be ingested, but they are not infective. The larvae develop into the infective fertile eggs after 18 days to several weeks. This development depends upon the environmental conditions. For optimum growth, it requires moist, warm and shaded soil. After infective stage eggs are swallowed, the larvae hatch, invade the intestinal mucosa, and they are carried via the portal and then the systemic circulation to the lungs. The larvae further mature in the lungs. This happens in 10 to 14 days, penetrate the alveolar walls, as in the bronchial tree to the throat, and are swallowed. And upon reaching the small intestine, they develop into adult worms. Between 2 to 3 months are required from the ingestion of the infective eggs to oviposition, that is the egg production by the adult female, and the adult worms can survive in the intestine or live up to 1 to 2 years. So, how do you clinically diagnose, or what is the manifestation of ascariasis? People infected with ascaris often show no symptoms regardless of the species of the worm. If symptoms do occur, they can be light and it will include abdominal discomfort. Heavy infection can cause intestinal blockage and impair growth and malnutrition in children. Laboratory diagnosis. One is microscopy. The standard method for diagnosing ascariasis is by the identification of the eggs in the stool sample using a microscope. In very light infections, when the worm load is less, eggs will be difficult to find under microscopy. So, normally concentration procedure is done before visualization of the eggs in the microscopic methods. The second aspect is serology. During the pulmonary disease, the larvae will be found in the fluids which is aspirated in the lung, from the lungs, and the WBC cells or the white blood cells counts may demonstrate peripheral eosinophilia because of the presence of these parasites in the lungs or in the alveolar cells. Treatment, prevention and control. Albendazole and mebendazole are the drugs of choice for treatment of ascaris infections. Use of properly functioning and clean toilets by all community members is one important aspect of, and also proper hand washing. These are the two main hygienic practices which can control the spread of infection. Eliminating the use of untreated human fecus as a fertilizer is also equally important. In areas where more than 20% of the population is affected, mass drug administration is normally advocated. That is, irrespective of whether the person has been infected with the pathogen or not, the whole area, when 20% of the population is affected, community medication, mass drug administration to the whole set of people who are living, the population who are living, in specific dosages required to clear them of the helminth infection. That is called as mass drug administration, is normally done. Anisakis. The worm is often referred to as herring worm, seal worm, cod worm, or the whale worm. This is an invasive larvae and it commonly infests populations consuming raw fish, such as in Japan and Netherlands, where raw herring is often consumed. The life cycle involves warm-blooded sea animals, and human beings are accidental hosts. So, humans is the accidental host, as I said. You have the marine mammals which contain the L3 to L4 larvae, and the adult final form is also seen in marine mammals like dolphins, whales, etc. In defecation, the free eggs float on the ocean. Now, the free eggs molt and generate L1 to the L3 stage larvae. These form the embryonated eggs. The eggs hatch. You have the free living L3 larva which floats in the marine environment, which is infested by or ingested by the crustaceans. The crustaceans are the intermediate and the transfer host in this case. The crustaceans are ingested by sea fish, cephalopods, and the higher organisms in the food chain. So, they get infected, and humans, these are again infested by the other larger marine mammals or marine organisms which also get infested. That is how the life cycle is completed. As I said, human beings are the accidental hosts. Anisakiasis is often diagnosed when the affected individual feels the tingling up of the worm. Acute abdominal pain, similar to acute appendicitis, is accompanied by a nauseous feeling. Symptoms appear 1 hour to about 2 weeks after the consumption of raw or undercooked seafood. With their anterior ends, these larval nematodes from fish or shellfish usually burrow into the wall of the digestive tract to the level of the muscularis mucosae. Occasionally, they penetrate the intestinal wall completely, and they are also found in the body cavity. Anisakids usually do not reach full maturity in humans. So, as I said, humans are accidental hosts. Normally, this is seen in the marine environment, completing the life cycle among the various sea animals, and they are eliminated spontaneously from the digestive tract lumen within 3 weeks of infection. So, if you look into this picture, see, you have the diagnosis of their elimination can be made. So, this is the first stage, which are the definitive hosts. They will excrete unembryonated eggs in the fecus. In stage 2, the eggs will become embryonated and L3 stage larvae are formed. These L3 stage larvae which are formed in the egg now will hatch as an ensheathed free swimming form. The free swimming larva is consumed by crustacean, and in the crustacean hemocoel they will develop. Now, these crustaceans which are infected are eaten by fish or these other cephalopod parentenic hosts. The L3 larvae are released in digestion. They will migrate to the mesentery muscle tissues, and they can be transferred to other hosts via predation. So, once they have been transferred to the other hosts, these maintain the L3 larvae in the tissue, and they are now these are infective, and they will pass on to the big marine mammals like the definite hosts, fish, squid, etc. All these are the definitive hosts. They again inside the larva. So, if a human being consumes this undercooked food, then they will get transmission of the particular food via raw and undercooked seafood consumption. Anisakiasis involves acute abdominal symptoms, usually within hours after ingestion of the larvae. This non-specific abdominal distress can be mistaken for other conditions like peptic ulcers, food poisoning, and appendicitis. Occasionally, the larvae are coughed up. Most infections are self-limiting as the larvae are unable to survive for long periods in the human host. But the associated tissue damage can cause longer lasting symptoms. Antigens then remain in the fish mussel after the larvae are killed. That is, the fish after freezing can cause allergic reactions in some individuals. A company called as AZTI has developed a molecular method that is able to identify the presence of Anisakis simplex, the organism, in fresh, frozen and processed fish products like smoked and cooked fish, etc., which are stored in a matter of minutes. This is a very rapid method because all the other methods for the detection of this pathogen take hours together by the other methods. So this is one, even in the frozen fish or including the fresh sample and including the frozen sample, if the food is contaminated by or the organism, the contamination can be detected by this particular method devised by AZTI. This method is based on DNA analysis by real-time PCR technology to detect the presence of Anisakis in fresh, frozen and processed foods. And Anisakiasis can be easily prevented by adequate cooking at temperatures greater than 60 degrees centigrade or by freezing. The FDA recommends all shellfish and fish intended for raw consumption be blast frozen to minus 35 degrees centigrade or below for 15 hours, or be regularly frozen to minus 20 degrees centigrade or below for 7 days. Salting and marinating will not necessarily kill all the parasites, as in Italy, where 2/3 of the cases were attributed to the anchovies marinated in lemon and vinegar. So, normally salting and marinating is a method to control microbial growth, but in this case it will not prevent infection because there was a epidemic which is reported in Italy where cause of the anchovies. The next parasite we are going to talk about is the tapeworm. These worms have complex life cycles and are relatively high order of the species specificity. The three common types are Tinea saginata found in beef, Tinea solium found in pork, and the Diphyllobothrium latum which is found in fish. Tapeworms have been known to reside for many years in the human intestine or in the animal intestine and can grow to very great lengths. Reproduction is ultimately sexual, but adults have both sex organs. The tapeworm inhabits the intestine of the hook by fastening itself by the head to the lining of the host intestine. Anatomically, the worm is composed of a head which is called as a scolex, a short neck and a tape-like body which is called as the stobula. The stobula is made up of segments. Each segment is called as a proglottid and it is linked together by muscles and nerves. And each proglottid absorbs nutrients from the contents of the human or the host intestine. The eggs are released into the lumen and they are shed. Some individuals have been known to harbor tapeworms for up to 30 years. And problems occur if the worm obstructs the intestinal tract. Eating undercooked meat from infected animal is the main cause of tapeworm infestation in people. The risk of complications depend upon several factors, including the type of tapeworm and whether or not the patient receives treatment. One is the cysticercosis. If a human ingests pork tapeworm eggs, then there is a risk of the larvae infection. The larvae can exit the intestine and infect the tissues and organs elsewhere in the body, resulting in lesions or cysts. Second is neurocysticercosis. This is a dangerous complication of pork tapeworm infection. The brain and the nervous system are affected. The patient may have headaches, vision problems, seizures, meningitis and confusion, and in very severe cases the infection can be fatal. Echinococcosis or the hydatid disease. The Echinococcus tapeworm can cause an infection called as echinococcosis. The larvae leave the gut and infect organs, the most commonly the liver. The infection can result in large cyst which place pressure on the nearby blood vessels and this affects the circulation. And in severe cases, surgery or liver transplantation is required. So, this is the life cycle of the dog tapeworm, see, normally you have the flea, which is an external parasite on dogs. The flea is in the environment, ingests the egg before they jump onto the dog. Now what happens is that the dog eats a flea which carries the tapeworm larva. So you have the adult flea on the dog, which this dog eats. The tapeworm larva grows into the tapeworm in the dog intestine, and this is now shed. Then segments of the tapeworm which are secreted get shed. So this cycle will continue. In this lecture, we saw some of the common parasitic infections which are foodborne, their prevention as well as the treatment. The question posed at the end of this lecture is, what are the common precautions to be taken to prevent infection by foodborne nematodes? Thank you.